यस्य रूपमधिकम् प्रच्छन्नगुप्तं धनम् विद्याभोगकरी यशः सुखकरी विद्या गुरुणा गुरुः विद्याबन्धुजनो विदेशगमने विद्या परं दैवतम् विद्याराजसु पूज्यते न तु धनम् विद्याविहीनः पशुः विद्या प्रशस्यते लोकैः विद्या सर्वत्र गौरवा विद्यया लभते सर्वम् विद्वान् सर्वत्र पूज्यते ज्ञानेन सदृशं पवित्रमिह विद्यते न हि ज्ञानेन सदृशं पवित्रमिह विद्यते न हि ज्ञानेन सदृशं पवित्रमिह विद्यते
Dr. Vishal Kadu is an academician and researcher with a profound expertise in molecular biology. Holding an MSc in zoology from the Department of Zoology at Savitribai Phule Pune University, he has demonstrated academic excellence by clearing the CSIR-NET examination 6 times with top ranks alongside clearing the Maharashtra-SET and GATE examinations. Currently, Dr. Kadu leads as the head of the Department of Zoology at Sathaye College Autonomous and serves as the controller of examination, bringing over 11 years of teaching experience to his roles. Dr. Kadu's research contributions are both diverse and impactful, spanning model organisms such as Hydra, Drosophila, giant wood spider, African giant snail, and Indian bats. His professional journey includes research tenures at esteemed institutions like the Agharkar Research Institute, ARI, Pune, and the Indian Institute of Science Education and Research, IISER, Pune. Notably, he has published over 33 gene sequences in the NCBI GenBank, authored 7 research articles, and authored 4 books on subjects including Instrumentation and Biotechnology, Genetics, Molecular Biology, and Biochemistry. In addition to his scholarly pursuits, Dr. Kadu has successfully completed 2 Mumbai University-funded minor research projects and is a recognized PhD guide in zoology. His dedication to fostering research among undergraduate students is evident in initiatives such as the Research Journal Club and research internships. Dr. Kadu has also contributed to student development as a judge in the Agharkar Research Competition held across universities in Maharashtra. Let's welcome Vidyvchspati, Dr. Vishal Kadu.
Hello, I welcome you for this another session of Vidyvchspati, where I will be briefly discussing about my PhD thesis. The title of my PhD thesis is Molecular Characterization of Tyrosine Catabolic Genes in Indian Bats. My research guide was Dr. Ashok Manekar from the Institute of Science, Department of Zoology. For this particular research project, we chose bat as a model system for certain advantages. Bat, being mammal, shares many characteristics with human, and many of the metabolic pathway that are there in the bat, they are known to be similar to that of humans. In this particular work, we discuss about tyrosine catabolic pathway, for which many diseases they are known in human. due to the genetic defect or mutations in the certain gene. When it comes to bat, they are comprising around 20% of the mammalian population throughout the world, as well as there is a lot of information available about the number of species that are available. More than 1100 bat species, they are already been identified and reported. Bats, they are the true flying mammals. They are found throughout the globe. They are very important as far as the pollination of plants is concerned, and they also keep check on the insect predator. Bats, they belong to the order Chiroptera, which splits into two suborders, Megachiroptera, which comprises megabats, or they are also called as fruit and nectar eating bats. Then another suborder is Microchiroptera, which makes a group of microbats or insect eating bats. A small group of these bats, they also have a vampire ability, that means they feed on the blood. As far as the diversity of the feeding is concerned, bats, they may feed on insects, they are called as insectivorous bat. Few bats, they may eat other small mammals, so they are categorized under carnivory. Few bats, they feed on fishes, so they are called as piscivore. Then few bats as vampire bats, they feed on the blood. And then another category of bats which feeds on the fruits as well as nectar. Needless to say, over the period of time in the evolution, all these bats, they have developed a significant adaptations so as to fit with their ability to choose the food. Microbats, they mostly choose their food depending upon the echolocation cues, and the megabats, they mostly use olfaction and vision to detect their food. In addition to this, few bats who could use visual cues to detect the food, and they could capture it in the light as well as in total darkness. Bats, as they have adapted for different kind of food, they also have shown the adaptation with respect to their digestion of food. For example, the bats which are feeding on the food like fruits, they have the gut flora which is adapted to metabolize the fruit compounds, as well as some insectivorous bats, they have a microflora which is adapted for eating the insect exoskeleton, which contains chitin, which generally is difficult to digest. So over the period of time in evolution, these bats, they also have shown the adaptation for the digesting the material which they ingest. When it comes to tyrosine is basically one of the amino acids which is synthesized by another amino acid called as phenylalanine. To make this conversion, phenylalanine hydroxylase enzyme is necessary. In addition to that, when the proteins, they are worn off, they are hydrolyzed to generate number of amino acids. One of the amino acids is obviously tyrosine. So this tyrosine can be utilized to make another molecules like adrenaline, non-adrenaline, dopamine, important hormone like thyroid hormone, and melanin pigments. But when the tyrosine amount gets in excess, it is been catabolized by a tyrosine catabolic pathway which involves 5 different enzymes. Tyrosine catabolic pathway, which involves number of genes, they produce the enzymes, and these enzymes carry out the metabolic pathway. As we saw, phenylalanine amino acid is converted into tyrosine with the help of an enzyme called as phenylalanine hydroxylase. Tyrosine further, when it undergoes a catabolism, it breaks down into 4-hydroxyphenylpyruvate with the help of an enzyme tyrosine aminotransferase, which is encoded by gene, we call it as TAT. The absence or mutation of this gene in humans leads to a condition called as hereditary tyrosinemia type 2. 4-hydroxyphenylpyruvate compound further catabolized into homogenticet with the help of an enzyme called as 4-hydroxyphenylpyruvate dioxygenase, that is HPD, encoded by a corresponding gene which we call it as HPD. The mutations in human leads to a condition called as hereditary tyrosinemia type 3 when the person has a defective gene or mutated gene. Homogenticet molecule further broken down into mallyl acetoacetate with the help of an enzyme called as HGA12 dioxygenase. This enzyme is encoded by HGD gene. The mutation in this gene in case of human leads to a condition called as alkaptonuria, which basically converts the urine or provides urine with the black color. The mallyloacetoacetate is converted further into fumaryl acetoacetate with the help of an enzyme called as MAA isomerase. This enzyme, when it is defective, it is seen that there are no reported diseases which are associated with it, but to complete the metabolic pathway, it is important to have this enzyme in place. Fumaryl acetoacetate compound further broken down into fumarate as well as into acetoacetate with the help of another enzyme called as FAA hydrolase, which is encoded by FAH gene. The defective gene in case of human leads to a disease condition called as hereditary tyrosinemia type 1. With all this metabolic pathway, what we are trying to emphasize is that the tyrosine is essential molecule not only for making number of biological molecules, but it is essential to have a breakdown of this amino acid. In this particular study, our hypothesis was that there must be having some form of connection between the type of food that bat are choosing with respect to the amount and the adaptation of these tyrosine catabolic genes. We hypothesized that the bats which are using protein rich diet, that is insectivorous bats, they must be having high level of these tyrosine catabolic genes as well as their enzymes compared to the fruit eating bats which generally have low amount of protein in their diet. For this particular study, we chose one frugivorous bat, we call it as Cyanopterus sphinx, and we chose two insectivorous bat called as Hippocidus fulvus and Scotophilus heathi. At the time of study, all three bat species, they were under least concern category as per IUCN data. In case of analysis of this hypothesis, we first got the permission from Maharashtra-SET Board Biodiversity Board, and whatever process we followed during the experimentation, they are as per the guidelines of CPC-SEA. So all three species of bat, they were collected from Elephanta Caves, Mumbai. Once the bats were collected, they were morphologically identified using the features like ear pinna, size of nostril and size of head. After identification of bats, the liver tissue was isolated. Then from this tissue, the whole mRNA was isolated. mRNA was further converted into cDNA, and then this cDNA was amplified using a gene specific primers. We used 4 tyrosine catabolic genes plus 1 housekeeping gene for all the 3 bat species under consideration. After the PCR was done, the PCR product was purified, and then the DNA sequencing was done using Sanger's method. The data generated was further analyzed using bioinformatic tools. On other side, we started with liver tissue. We isolated whole mRNA, converted that mRNA into cDNA, and then we performed real-time quantitative PCR just to find out the quantity of RNAs synthesized by different bats in the liver. Here for this study also, we have used 4 tyrosine catabolic genes plus 1 housekeeping gene for each bat under study. Then the data was analyzed for the research purpose. The method involves serial analysis of gene expression, which basically tells us about how the genes are expressing across the different species. For this particular study, we proposed the housekeeping gene, which is eukaryotic translation initiation factor 2, where we are the first to propose this particular expression of gene in the liver of bat species. Before we analyze the data or before we do the quantification, we normalize the level of this housekeeping gene. For the purpose of bioinformatics analysis, we used softwares like Clustal Omega and Clustal 10. The primers were designed with the help of software called Primer 3. For phylogenetic tree, we used Bayesian version 3.1.2, and to make the phylogenetic tree with particular conditions, we used neighbor joining tree method using Mega 4 software, where the Kimura 2 parameters were used with 2000 bootstrap replicates. This particular image shows the isolation of RNA. On left hand side we have RNAs of Hippocidrus fulvus, then at the middle we have DNA ladder, on right side we have Scotophilus heathii RNA. This particular table basically explains how the genes are expressed across the species that were under consideration. On left hand side we considered frugivorous bat and tried to compare its expression with the insectivorous bat. So as far as the expression of housekeeping gene is concerned, we normalized it across the species. For Cyanopterus sphinx and Hippocidrus fulvus, we saw the upregulation of genes in all 4 genes under consideration. When we did the analysis of Cyanopterus sphinx with the Scotophilus heathii, again we came up with the same observations that there is upregulation of genes which are involved in the tyrosine catabolic pathway. When we compared the 2 insectivorous bat species for the tyrosine catabolic genes, we saw the expression were significantly same. This particular data tells us about the gene sequences that are already been published in NCBI GenBank for all the 3 species that we have used. This particular data tells us about the sequence comparison for nucleotide as well as for amino acids. In the above table, what we see, we used Cyanopterus sphinx and compared it with Hippocidrus fulvus as well as Scotophilus heathii. So in nucleotide sequence for eukaryotic initiation elongation factor 2 as well as TAT gene, we saw there is a significant similarity and more than 90% nucleotide sequence is matching. When it comes to HPD gene, the sequence similarity was slightly reduced when it compares between the insectivorous and frugivorous bat. But there was significant similarity across the Hippocidrus fulvus and Scotophilus heathii. For HGD gene, we saw again it shows similarity across both the category of bat species. For FAH gene, there was significantly reduced amount of similarity as far as nucleotides are concerned across all 3 species. Basically, what does this indicate? That there is some possibility of having conserved nucleotide sequence across the species, but there are few genes where there is a variation as well. When we compared the amino acid sequence for each one of these genes, keeping the comparison type same, what we found that in case of insectivorous bats, the sequence similarity is on the higher side, which is highlighted on the right side. More than 90% sequence similarity was seen for nucleotide for all the tyrosine catabolic genes. The phylogenetic tree analysis for eukaryotic elongation factor 2 basically explains us that Cyanopterus sphinx and Scotophilus heathii, they have had a common ancestor recently, and this ancestor must have split from Hippocidrus fulvus bat species. For this particular species, we used mus musculus as an outgroup. For phylogenetic tree of HPD gene, which says that the Scotophilus heathi and Hipposidrus fulvus, they have had common ancestor, which was split from another bat species, Cynopterus sphinx. In this case, again the outgroup was used, the mus musculus. Rest of the species that we see in the phylogenetic tree, they all are bat species. This basically gives us the lineage and the evolution of the bat species, where we could see the feeding habit must have evolved differently for different bat species. That means across all the bats we do see there are few bats who are frugivorous, there are few bats who are insectivorous. For TAT, HGD and FAH gene, what we could see is this: Cynopterus sphinx and Hipposidrus fulvus, they are having a common ancestor which is diverged quite some time from that of Scotophilus heathi. This particular diagram helps us understand that the evolution of all these three species have conserved for TAT, HGD and FAH gene. For eukaryotic elongation factor 2 gene analysis, which is used as a housekeeping gene, before doing the quantitative PCR, the levels of eukaryotic elongation factor 2 were normalized so as to ensure that all the three species that are under consideration, they have same level of housekeeping gene. Then it is known fact that this particular gene expresses in the liver in the constant level. That means all the cells of liver, they generally express this particular gene. Whatever differences that we see in this eukaryotic elongation factor 2 gene across three bats, that could be due to synonymous substitution, because though there are nucleotide differences, there are only 3 amino acids which shows variation across 3 different species. That means eukaryotic elongation factor 2 gene must be conserved evolutionary. Then we also see that Cynopterus sphinx and Scotophilus heathi are genetically more related to each other compared to that of compared to the fulvus species. TAT gene, it is known that for tyrosine catabolic pathway, the TAT gene acts as a rate limiting enzyme. We saw there is a significant upregulation of TAT gene in insectivorous bat. This also proves that the bats, being insectivorous, they are having protein rich diet, so they will be getting more amount of tyrosine, which is broken down by this rate limiting enzyme. We also observed that there is purifying selection for insectivorous bats. That means the evolution has permitted least changes in tyrosine catabolic gene for insectivorous bat, but opposite to that there are quite a few variations we see in case of fruit bats. That means there is a relaxation kind of evolution. TAT gene have evolved multiple times during the evolution as per the phylogenetic tree. Most of the substitutions that we observed, they were type of synonymous type of substitutions which are supported by bubble hypothesis. For HPD gene, there is manifold increase in the transcript of insectivorous bat. Then there is a more sequence similarity as far as HPD gene is concerned amongst the insectivorous bat compared to frugivorous bat. The nucleotide differences of HPD for frugivorous and insectivorous bat could be due to non-synonymous substitutions, which has reflected into the amino acid variations across these two categories. Insectivorous bat shows the separation from each other after splitting from frugivorous bat as far as HPD gene is concerned. Then we have certain positions like amino acid position number 34, 110 and as listed. These positions could work as a hotspots for mutation. HGD gene is known to have 14 exons and it is commonly expressed in the liver, then kidney and prostate tissues. As far as the level of HGD was concerned, HGD mRNA is concerned, there was observed a many-fold increase in the mRNA level with respect to insectivorous bat. Relatively, the more nucleotide sequence conserved was seen in case of insectivorous bat compared to the frugivorous bat species. Out of 25 nucleotide substitutions that were observed, 18 were synonymous substitutions and 7 were non-synonymous substitutions, which ultimately has resulted in the alteration of 5 amino acid. We also found that there was no insertion or deletion in the HGD gene when we compared the sequence across the species. For FAH gene, we saw the many-fold increase in the mRNA level of insectivorous bat compared to frugivorous bat. Cyanopterus sphinx and Hippocytrus fulvus are more conserved with respect to amino acid sequence as well as nucleotide sequence when it compared to other species. Out of 68 nucleotide differences that we observed, 40 nucleotide shows synonymous substitutions and 13 nucleotide shows non-synonymous substitutions. Synonymous substitutions basically allows this gene to passed on to number of generations without having any deleterious effect. After doing all this work, we made certain observations, and they are the variable expression of tyrosine catabolic gene across the insectivorous bat and frugivorous bat. Eukaryotic elongation factor 2 being housekeeping gene can be used as a control for studying the variation in the gene expression. Then, in case of phylogenetic study, tyrosine catabolic genes suggest to have evolved early for frugivorous bats than compared to the insectivorous bat. Then, as far as tyrosine catabolic genes are concerned, all those genes under study, they show some form of conserved regions. That means these sequences, they matches across 3 species that we used. Then feeding habit and diet content of these bats, they directly have a correlation with the amount of tyrosine amino acid and the level of enzymes which are associated with it. That means the insectivorous bat will have high amount of expression of these tyrosine catabolic genes. We also observed that there is a relaxation of evolutionary constraint for TAT, HPD and HGD gene for the frugivorous bat, and some amino acid substitution that should have adverse effect into the enzymatic activity. Fruit bats have reduced their reliance on the tyrosine catabolic pathway genes for a reason that they might not be needing this enzyme in higher quantity as otherwise it is needed by insectivorous bat. During our study, we did not find any evidences of gene duplication with respect to the pathway. So we believe that the bats, they have single copy genes for this tyrosine catabolic pathway. I thank you for your attention.
ज्ञानेन सदृशं पवित्रमिह विद्यते ज्ञानेन सदृशं पवित्रमिह विद्यते ज्ञानेन सदृशं पवित्रमिह विद्यते